record ko na lang kapan. So this is our course, and in your senior high school, nag-senior high school man mo tanan, diba? So senior high school ninyo naamoy study of kanang genes, so or genetics. So you have you should have prior background to the study of DNA and genes, not only in animals, no, or humans, in higher animals like humans, but also in crabs. So we studied this subject for improvement of our crops and animals for livestock and also food production. So therefore, we will talk about as a whole mga genetic processes, developments, biotechnology, and genetic engineering. So this will all be explored in the subject. So these are our course objectives para meron kayong knowledge and skills about the latest technology on how to improve, no, not only in crops but also animals, but in humans it's also part of although human cloning na talaga, we just don't know, but it's not really legal kasi it's unethical. So right now hindi patanggap whole worldwide ang human cloning. So can you imagine, no, na human clone? So lisod pa siya tanggapon, di ba? But it has been a prophecy since before na moabot pa gyud ta nga panahon, moabot ta nga technology. But right now, we are still in the middle. So we and also mga simple techniques and about genetic processes, biotechnology, and genetic engineering. Although wala man tay lab, but tissue culture perhaps is already part ng biotechnology. And also most importantly, to appreciate how to improve our crops and animals for the benefit of the human race. Anyway, so again, we all know our vision, mission, goals, course, values already. So let's move on to the course outline. But I will give you the schedule of learning activities. So sa ating first week, we are supposed to have yung introduction, or kasali na dito yung history ng biotechnology, yung importance ng adoption, and about plant breeding and animal breeding genetics. And after a month, sa module 2 na tayo. So ito yung coverage ng ating prelim, mga techniques in crop improvement. So medyo very lengthy and bulky ang ating coverage. So as much as possible, paspas sa dapat, but maybe every 2 weeks, kasi marag ako na ning ihap ang topics every 2 weeks na ako ay duha ka assignment isend sa inyo. In the later mga finals, tag-isa na lang ka topics. So there are actually 18 supposedly topics that you're going to answer. And then 3 exams, no, for the midterm, pre-final, and finals. So tissue culture, DNA extraction, and gene coding. So although dili na ninyo ma-perform, but you should also try to, kasi naman na siya maraganig sa inyong exam na mga coding. So actually sayon ra kay na tagaan ta na mog tip daan, i-search lang na ninyo ang kanang supposedly unsa iyahang kasi ano man na siya definite and standard kung unsay mga coding nato sa tong genes, like the 22 or 21 amino acids. So naka-code na nga mga genes. In fact, ang humans are already coded, and some of the plants like a lot, no, mga wheat or katong pinakauna nga ginastadihan nato nga plants, I just forgot the name, but ma-mention na sa tong mga lesson. And also animal DNA extraction and so on. And then we are going to talk about also GMO, genetically modified organism. So, so far, right now, we are actually consuming na mga GMO, no? Whether we like it or not, but although we have the freedom to choose kung alin, but sometimes dahil hindi labeled yung food, especially kung wala tayo proper nga mga regulations sa atong country, but we already have, which will be talked in the last weeks, no, about laws and regulation in biotechnology. And also issues like why do you agree with GMO and why not? No, these are the issues that we need to discuss, aside from the different amazing GMOs that had been created and also commercialized already in the country, like yung Bacillus BT corn. Have you heard of BT corn? Nakadungog na mo?
Wala, ma'am.
Wala pa?
Good, ma'am.
Sige, karon makadungog na siya. So, and also, and yeah, so these are our activities. So usually mag-asynchronous task through 40-minute lecture lang or consultation or tutorial kagaya nito. Unfinished usually kasi 40-minute lang ta, and you will spend the rest of your time kasi 5 hours ata per subject ito and per week. So sa remaining time ninyo, which is 4 hours, you will be directed permanently sa inyong quiper and to your laboratory exercises na hindi ko pa na-send, no? So ipakita ko sa inyo ngayon ano yung laboratory exercises. And part of your assessment are yung mga quizzes na nasa quiper. Actually 20% lang siya, but usually gina-change na ako na in the end, no? Laboratory exercises ninyo na report ay 40% at yung total ninyo preliminary final exams ay 40% of your grades. So, ano ito yung mga quizzes yung nasa Quiper. And then yung exams, isa-send ko rin through Quiper essay. So, familiar naman mo sa Quiper essay, no? So, ang pag-answer sa Quiper essay. So, musulod naman ko na sa inyong account. So, ito, andito na lahat yung course pack, no? From the lesson study guide 1 hanggang study guide 2 by module na siya. So, nandito na lahat ng lesson, but more on, ano kasi ni siya, more on description lang. So, lahat ng mga animation and, you know, presentation are in other form ng PowerPoint din na makikita din siya supposedly dito sa ating Quiper. Tanawa, no? Refresh pa. So, what is our, so ako pa din siyang isahon lang na ako ni pag laboratory exercises para kabalo na mo ang sa inyong buhaton. So your first laboratory exercise is actually demonstrate ninyo ang monohybrid and dihybrid cross using the Hornet square. So ano yung Hornet square? So naana siya sa ating lessons about the study of genetics. Wala pa na ako na-send sa inyo, but you can always search what is a Hornet square. Dali lang man na siya, i-google lang na ninyo, no? And pwede rin na inyo ma-memorize, but still, naka-remember mo sa inyong biology before about Hornet square? Ka-remember mo something like that? Wala? So, wala? So this is wala. So this is an example of a Punnett square, no? So whether ano siya kanang sa tawag monohybrid or dihybrid cross siya. No, imuha lang na siyang basta nana siya ginatawag na heterozygous, homozygous, and dominant and recessive. So for example, mo father side, mother side. So kuan lang na siya kanang monohybrid kasi meaning the same lang siyang isang ano lang siya, isang tawag na isang characteristic, isang genetic makeup. So pag i-pair lang na nimo siya, i-pair lang nimo siya ana. So that means capital ang dominant, small letter ang recessive, meaning pag dominant meaning halim for example, 'di ba? Like anang imuhang parents kulot og straight og buhok. Tapos ikaw straight imong buhok maski imong papa kulot. So it means ang straight nga buhok ang dominant, no? Like that. So but then ang kulot nga buhok na agyapon na sa imuha, no? And sa imong other siblings, sa imong other igsuon, pwedeng wala, pwedeng naa kasi BB siya, no? Capital dominant B. And so on. Anaon na po na siya, no? Small letter B and capital letter B and small letter B. So i-pair lang na nimo na siya sa square. So that's Punnett square in advance. Lagi ko. Okay, so that's your laboratory exercise 1. And ako pa ni siyang isend sa inyo sa paper, ha? Wala pa lang karon. And then you are going to perform also DNA extraction or tissue culture at home using mga household na ano. So actually na ako'y ano, but there are a lot. Pwede ring kamo na or tagaan tamo og home tissue culture na media, home tissue culture, whatever. So daghan kayo siya references about home tissue culture. Larawan man lang siya. So nga mga ano, pwede nyo buhaton, but then maybe I'll just give you what to do, no, an instruction para maka-practice mo. But if ever maka-add to ta og isa ka tissue culture lab, the better, no? Someday kung magkaroon pa ng enough time. So that is just an overview of what you're going to do with your laboratory exercise. So those are all home-based. Ibig sabihin, you're going to do that individual at your own time, at your own resources. So pwede ring, kasi at this point, dili pa ta pwede jud magsulod sa classroom. But then we're planning na nga mag face-to-face. Not limited lang siya. But sa ato lang mga laboratory para every laboratory pwede na mumusulod sa school para ma-perform, like makagamit og microscope, makagamit og mga lab equipment. So sa next sem pa noon, not this semester. So may naay plano ang admin na sa sa summer class. So most probably you're going to have summer class. So hopefully madayon ang atong maraganig trial. Mark trial pa nato. So that's your laboratory exercise and your course topics for this subject. Do you have any questions or clarification? So ayan sa wakas, no? But murag 61 pages ni kasi siya. So i-click lang o click ninyo balag di nyo basahon kasi kuan man pugod ni siya kanang the whole kuan na gini course guide, the whole subject na gini. Like this one. No, marag study guide na niya siya, marag whole lesson na gini sa whole subject. So i-click lang sa ninyo ni siya, bahalag di pa ninyo ni siya basahon, no? Kailangan maklik na ninyo para mag-register siya nga complete, and then you answer the question here. Okay, so naamay mga questions or clarification before we proceed with our first topic? Question or clarification?
No, ma'am. None, ma'am.
So klaro ba tanan ang sa inyong mga requirements ang gina-expect na buhatonon? Klaro na? Okay, so sa exam ninyo is always open notes and also work from home. Kanang home assignments lahat, even laboratory. So that would be easier for you. Unlike, it's kasi the same question lang po akong ginahatag for for face-to-face. So, it would be easier for you. Kaso lang, medyo mahaba man good siguro to akong mga exam. So, tag 100 items most probably. So, i-send ako na siya after a month for your prelim. And you have actually the whole SEM hanggang January 30 to, actually January, January, sorry, June 20 ang ato ang finals. So, but then extended ang nakabutang na ako nga dates hanggang 30. But hopefully, you're going to finish as early as possible. Okay, so wala na yung mga pangutana?
Wala na, ma'am.
So, pag send na ako pa mag-uni arrange. So, but most probably, mo to yung una buhaton sa inyong laboratory exercise 1, which is on, actually, naapod ni, okay. Wala na ako siya gibutangan pala ng video. So, naapod ni siya video kasi sa classwork na ako yung video. I mean, sa Google Classroom. Na recorded video ni siya last time. So ako lang po ni siya palang i-add para for those who miss kasi although na-upload ko ikuan karon, recorded pud ni ato ang session. So i-add nang nako ang duha ka video para sharo na jud. So in here, asan ako? Oh na. Asa kinalas ay nagyud ay kigi-post nga video. So in the end na ako gi-post nga video nga tan-awon nato, tan-awon ninyo pod, no? For the whole lecture na siya. No, para di na ko sige balik-balik og lecture sa inyo sa first. So 10 minutes na lang ta. So do you have any questions or clarification before maubos ang 10 minutes natin? Regarding the topic. So the first topic we have is biotechnology applications and status. So mao ni ang history niya, what happened. So wala, so siguro padayon na lang ta sa video. Let's try to check.
Okay, good afternoon once again, and welcome to your class in ATC 111, Crop and Animal Improvement. So last time, we just have our orientation. Good afternoon, lovely. Can you hear me?
Yes, ma'am. Good afternoon, ma'am.
Okay, so were you around last meeting?
No, ma'am.
Okay, already I have posted your quiz.
Forward lang ako ha.
Your faces are specific.
Explain na
answered your quiz in advance. The issue that we have now is we call biotechnology. So in this presentation, you're going to know the different historical development defined by technology and genetic engineering process. And also, what is the global status of the biotech crops? We call them biotech crops or genetically modified organisms, or what we call as genetic engineered crops or animals. So I bet that you know that since civilization began like 2,000 years ago, so they say the earth or civilization have existed about 2,000 years ago, and along with that is for hunting and gathering. So we call them pastoralism. pastoralism. So definitely, they say that biotechnology is connected to years back when we do hunting and gathering. So humans as and early ancestors existed as food gatherers and hunters. So and when about 8,000 before Christ, the domestication of animals and the selection of plants began actually 8,000 BC or before Christ. So the Egyptians uh, master actually the art of wine making and baking leavened bread about 4,000 BC. So no una-una kasi we'll say this is for they call this Mesopotamia or in Egypt itself. We all know that the Middle East is the cradle of civilization. So dun kasi nag-start ang civilization. So where it doesn't mean naman na doon nag-start ang civilization, wala pang tao sa Pilipinas. It's just that, well, maybe yes, wrong, or not. So, but one thing for sure is wala kasing records historically na meron ng mga what specific time meron ng tao sa Pilipinas, no? And of course, based on your knowledge, when does the Philippines discovered? Based sa knowledge ninyo. Based on your knowledge, when does the Philippines discovered? By Magellan? Or was it 1521? No, March 16, 1521. Kamot lang ka. Ka-remember ba ko? Ka-remember mo? So, let's just proceed with, because maybe because of your connection, you cannot. Mary Rose, okay ka lang?
March 1521, ma'am.
Oh, yeah, so, do you really believe na 1520, diba? Pero pag 1521, naanay mga tao, naanatay mga deity, naanatay mga what do you call that, kanang communities, no? Meron na tayong mga communities, but prior to that, there must have been Filipinos already or early settlers before 1521. So anyway, to continue, on 1760s, it was marked as the age of industrial revolution. The use ng mga mechanical implements where the humans have able to use, instead of hunting and gathering food, they now have the so-called machineries in agriculture and even crop milling. So that's 1760s. So of course, way far, wala pa tayong lahat niyan. And on 1860, there's already deliberate plant breeding and pollination of plants. No? So in 1860, meron ng we have able to breed plants through pollination. And in the 1960s to 70s, it was marked as a period of agricultural advance known as the Green Revolution. So why Green Revolution? Because instead of existing na mga crops and animals, we use high-yielding varieties. We have able to choose na what are these good varieties and even use fertilizers and irrigation water and pesticides to increase crop yields and significantly strengthen food security. So, ayan. So, it's known as the Green Revolution, wherein nakaila na sila o mga different breeds hybrids, fertilizer, irrigation, pesticides, and so on. And also in 1953, because that's on the 1960s and 70s, as well as on 1953, if you know the following scientists, Watson, Crick, Wilkins, and Franklin, they elucidate the structure of DNA. So that was the time that they have able to identify the structure of DNA. And again, when we say DNA, deoxyribonucleic acid, it's the blueprint, the genetic makeup of our crabs, animals. So
Sorry. Okay,
so
so
I just have
okay. So at this point, I'm going to pause it kasi we only have 1 minute left. And before maputol ang ating time, sorry kasi limited lang yun atong time. And also, kasi ang sa ano man na, and also para dili magas makuha yung time for your quiper, for your asynchronous. So the question is, kailan talaga ang birth ng modern biotechnology? Anyone? Yes, James? James?
Ako po, ma'am.
Yes,
Sa 1973 po, ma'am.
very good. So, nakinig talaga si James, no? And, yes, so, dito yun nag-start, no? But then, ang biotechnology. But when we say genetically modified organisms or GMO, nag-start siya since ano pa talaga, no? Since yung Green Revolution, no? Kasi breeding is a natural phenomenon. It's a natural occurring